It's a pleasure to welcome Dr. Marco Bortolato to give his MKRC seminar today. I've placed a link in the chat that will allow you to go to his faculty page at the University of Utah to explore more about Professor Bortolato. Our paths crossed at University of Kansas a few years ago, and I got to tell you that the energy he brought to the department was amazing in terms of the level of grant getting activity and the clinical relevance. I've learned so much from a lot of my colleagues, but one thing I learned from Dr. Bortolato is study something that is medically relevant, for crying out loud. His laboratory is currently involved in two major translational research projects. The first target of his investigations is the characterization of the molecular substrate of gene-environment interactions. In other words, he wants to identify which genes respond to environmental stimuli, interactions, and impulsive aggression in particular. The second aim of his research is the development of novel steroid and cannabinoid-based pharmacological tools for the therapy of impulse control disorders, Tourette syndrome, autism, and schizophrenia.
And if you guys are interested about this, I can just tell you that we got a JCI publication yesterday in which we found a new therapy that is based, in fact, on steroids for opioid abuse disorder. So those of you who are interested can look into it.
Beautiful. The long-term goal of his research is the development of novel agents for treatment of pathological aggression, autism-spectrum disorder, Tourette syndrome, and impulse control disorders such as pathological gambling.
Yeah.
Dr. Bortolato, it's so good to see you again. So
likewise.
Stage is yours. Take it away.
All right. Well, thank you so much, Jeff, for this great introduction. And I'm happy to talk a little bit about the first line that you mentioned. And that's, in fact, our studies revolves around the ideas of how we study the biology of violence, which is a very important problem, as we will see. We will talk in terms of aggression, and definitely greatly understudied. There are very few labs that specifically address this problem. So when we talk about aggression, okay, the first thing that needs to be recognized is that, of course, aggression is a physiological reaction. You see it as a fundamental, purposeful behavior that really enables survival. But of course, when it comes to complex societal organizations, so of course in human society, that can also become pathological. And it becomes pathological when it typically exceeds the intensity of the stimulus that triggers it. So it is disproportionate, right, to what has provoked it. And of course, it entails, as such, acts of criminal violence. And that's why we talk about violence in that respect. And inevitably, like everything in psychiatry, when it's pathological, it needs to, by default, pose some burden or a reduction of functioning at the level of the individuals, and of course, in this case, the victims. And I'm sure you guys are more than familiar with the fact that violence, of course, in the United States, particularly in terms of gun violence, is rising. I would actually say that in general, as an overall trend, there is a reduction, but acts of impulsive violence, so in this particular context, in fact, particularly involving gang membership or, for example, gun violence, are on the rise, as you can see from all these. I actually collected all this a few months ago, putting it all together. And it was really within 2 weeks, a lot of new episodes were occurring. And this is, of course, absolutely current today. And just to give you a sense of how prevalent this is, you can just look at, for example, the homicide statistics. And the United States, is actually number 4 in the list. And in general, I would say it is one of the countries in which we see one of the highest levels of violence. And as you can see, it tends to be overall quite well distributed across different states, even though there is probably some prevalence in the southern states, particularly among, I would say, low socioeconomic classes with low socioeconomic status. So just to give you a sense of the importance of this problem, these are relatively recent statistics. About 70% of our or violent crimes or offenses are due to pathological aggression. How do we know this? Because typically we profile convicts, and we see that in most of the cases they have some problem of impulsivity that has driven the specific act of violence. And generally speaking, the collective burden is really very high, is as high probably as what we see for substance use disorders, with which incidentally there is a lot of comorbidity. And we're talking probably, and this is probably an underestimation, about 200 to 300 billion per year, which is remarkable because, as I said, so little is done in terms of research, particularly biological research on the problem. So when it comes to pathological aggression, and we look at we look at the problem from a psychiatric prism, I would say there are several entities in the DSM-5, which is our manual or reference, of course. And you will see oppositional defiant disorder, conduct disorder, intermittent explosive disorder, or antisocial personality disorder. All these disorders in reality have a lot in common, and the level of comorbidity across these problems is extremely high. They tend to be differentiated primarily by a factor of age. This is in young kids. This is observed in adolescent, defined, I should say, in adolescents, and these are in adults with, I guess, a slight differentiation in terms of the specific characteristics of the aggression. But in reality, as we look at the biology of these problems, we see a lot of commonalities, a lot of overlaps. And I should also remind you that, of course, pathological aggression is extremely common in many other problems, ranging from PTSD, ADHD, bipolar, schizophrenia, of course, and substance use disorders. And it is, unfortunately, one of the core determinants of suicide, actual suicide, not just suicidal intention. So we see, for example, that 2 out of 3 victims of suicides have a history of high levels of aggression. And that's, in fact, aggression against the self. Still a very important form of aggression. When it comes to phenomenology, you understand that there is a huge spectrum that really that is encompassed by this problem, and so we look at a very heterogeneous condition. So clearly, whenever we have to deal with this from a scientific standpoint, we need some sort of, I guess, criterion to define how we can tease apart different types of problems. And I think the most important, for those of you who are interested, the most important criterion will be that of construct. So in which we talk about reactive versus proactive. So let's look a little bit more about what these two subtypes are. Essentially, when we talk about reactive aggression, we are dealing with primarily impulse control problems. So there is rage, hostility, and specifically one feature that you will see in most of these individuals is what we call hostile attribution bias, which essentially means that the individual that is affected by this problem tends to tend to misinterpret social cues. So for example, he might see another individual smiling and he might take this smile with a sort of bias, right? As if it was derision as opposed to friendliness. This is typically associated with a history of maltreatment. We see this quite a bit in individuals that have a history of child abuse. And that's typically enacted in response to frustration. So this is I would say, probably the most typical type of aggression that we see in the psychiatric practice, because some of these individuals realize that they are unable to control themselves and come to the psychiatrist. Slightly different and in a way more subtle and more problematic is the other subtype, the proactive aggression, which is what we typically associate with what we call psychopathy, or certain forms anyway of sociopathy. So these are individuals that tend to be aggressive, but in a very instrumental way. They do it because they have a gain. So this is not impulsive, this is premeditated, and it typically is associated with this callous and emotional trait, which is really one of the core features of this form of aggression that is premeditated and specific, and tends to have very low levels of empathy for the victim. Now, as I mentioned, these are two subtypes, but you shouldn't imagine that this is a dichotomy, right? There is a lot of overlap in between, and we really don't know why reactive, for example, we see it quite often that individuals might start as reactive aggressors and then gradually step into proactive aggression. This is very common, for example, in bullying. It starts from a pathological situation of children that have been abused, but they start becoming aggressors, and with a specifically premeditated goal. So we really are looking into these problems. When it comes to the circuit, and here I will talk primarily about reactive aggression because we know this much better, there are several areas in the brain that control this problem. And of course, the areas that we will primarily talk about today will be the prefrontal cortex and to some extent the ventral striatum. But let me just break it down for you. The prefrontal cortex is really fundamentally important because, generally speaking, in all reactive aggression, you have a low function of this area, which is very important for control, for behavioral control. So when this doesn't really work, inevitably you tend to have more disinhibited behavior, included aggression. And this is the primary, I guess, the top area of control of aggression. And then, of course, you have what we call a limbic subcircuitry, which is directly controlled by the prefrontal cortex, that includes the ventral striatum and the amygdala. This goes into the motivational factors of aggression. And finally, the effector circuit, and we won't talk too much about it, but this is quite important, that really mediates the behavior as a function of transmission from these areas. And this includes, of course, the hypothalamus and the periaqueductal gray. I want to delve a little bit more into the function of the prefrontal cortex because there are two main areas that communicate directly, primarily with the amygdala, which is located here, and that have both a top-down or a bottom-up control. So essentially, you can imagine that I guess the crosstalk between the different areas of the prefrontal cortex and the amygdala, and different areas of the amygdala, I should say, control aggressive reactions, particularly in response to how we interpret facial and social cues. So there is a consistent crosstalk that reevaluates what we see around us in terms of social cues, and then mediates aggression, the aggressive reaction. So just to give you a sense, and particularly I want to focus on the orbitofrontal cortex, because this is really important in appraising the motivational value and the effective valence of social stimuli. So imagine that we're dealing with the area that allows you to really understand whether you're dealing with individuals that might be threatening or not. And in fact, what happens in most reactive aggressors is that when we test them, for example, through this facial recognition, or I should say facial expression recognition task, even when the face is neutral, these individuals will tell us that they look angry, they look threatening. So again, that type of hostile attribution bias that I discussed earlier. So how is this coordinated at the level of the brain? There are many, many, many different factors, and we'll see some of them, but I want to, I guess, at least at this stage, really discuss one that seemed to be really fundamental in the regulation of aggression, and that's the serotonergic system. Serotonin modulates the crosstalk between orbitofrontal cortex and amygdala. Of course, both regions have ample projections from the serotonergic system. And the relationship that occurs in terms of the distribution of function, if you will, of serotonin across these two areas is really critical to shape our reactivity, our aggressive reactivity towards stimuli. So really important in appraising threat and responding to the threat. What do we do in the lab? How do we use this information? So what we try to do in the lab, as much as possible, is to use animal models, as Dr. Staudinger told you at the very beginning. We try to use really this idea of translating clinical ideas, clinical entities, clinical properties into animal models, and we study these intermediate phenotypes. So these building blocks, if you will, of a complex behavioral problem. And then inevitably we try to develop novel drugs based on what we see in terms of neural dysfunctions in our animal models. And then through this type of back and forth type of crosstalk across these different areas of our research, we ultimately developed drugs in the hope of, in fact, using them in patients. And we'll see a little bit of that. So what is the problem in this case? When it comes to aggression, I told you from the beginning that aggression can be physiological, and it very often is in animals, right? So when I take a mouse or a rat or any sort of animal, really, inevitably, in many cases, the aggression that we see is going to be physiological. So it's going to be actually positive for the animal, appropriate for survival, for example, for defense of territory. And so when we just look at a mouse, how do we know that aggression can be pathological or physiological? The reaction itself is pretty much the same. There are a few differences, to be honest, but they are not obligatory. So in many cases, they look absolutely identical. So how do we know that when we see aggression in mice, this is what the corresponding feature to pathological aggression as opposed to physiological aggression? And this is a big problem in the field because, of course, you don't want to reduce physiological aggression, which would correspond to appropriate behaviors in humans. You want to just address pathological aggression. So in order to do that, our lab has tried to look into the problem from a different perspective. So we just started looking at what is known in the field in terms of vulnerability factors. And as you can imagine, there are many genetic factors and many environmental factors. And I would say that the two major factors, genetic and environmental respectively, that drive aggression and antisocial behavior are from the genetic side, this MAOA, this monominoxidase A. This is the gene that consistently comes as a top factor in the list of genetic factors for aggression across many studies. And in environment, of course, poor child rearing, so neglect and abuse, child maltreatment. And I want to give you a little bit of an introduction on monominoxidase A. If you're more interested, we recently wrote a review and you can read more about the biochemistry of this enzyme. But essentially, this is an enzyme that serves, and this is not so surprising after all, the degradation particularly of serotonin. As you can see here, serotonin is degraded primarily by this enzyme. And this enzyme is also X-linked. So there is only one copy in males. And the reason why we started studying this problem is that in 1993, Hanbrunner in the Netherlands found a pedigree, I should say, several families, but interrelated, with extremely high levels of aggression, and he found that these were underpinned by a mutation, a point mutation, in fact, of the MAOA gene. And of course, it appeared only in males exactly for this reason. So that started a huge line of research in the field that, of course, led to many translational applications, including the generation of MAOA knockout mice. And as you can see, in comparison with wild type mice, these tend to fight quite extensively. They have exaggerated aggression. So even when they win a confrontation, they keep going at it. And that's, in fact, the connotation of pathological aggression that I described to you. And one of the most important features that I want you to remember at this stage is that these animals, of course, because they don't have a degradation system for serotonin, particularly in early life, have extremely high levels of serotonin in the brain. As you can see, this goes up to more than 5 times, and depending on the area, it can reach even 10 or more times in comparison with wild type. So essentially, the brain of these individuals is showered in serotonin at the beginning of life. Now, I won't go into too much. We studied these mice quite a bit, and we developed a lot of different tools to study what was happening, and we found out essentially that these animals seem to have autistic traits, which was really an important point because it was not known about the Brunner syndrome at the very beginning. But I will tell you, I want to go into this, but it's interesting just to, I guess, carry a point on what I was saying about translational strategies. We presented this article, and it was received with a lot of skepticism because, of course, this mutation was not associated with autism at the very beginning. But then a few months later, in fact, the first case of autism with the mutation of MAOA, with a point mutation of MAOA, was identified in humans. And in general, we do find a lot of resemblances between the clinical description that we see in Brunner syndrome patients and MAOA knockout. So they start with this type of social deficits that then become later on aggression when the individual reaches puberty, more or less. To just give you again an idea about this point, and this will be useful later on, we also studied the mechanism of these problems. And one major factor that we found was the implication of a very important receptor in the prefrontal cortex. Remember that I told you, generally speaking, impulsive aggression is linked to a deficit in the prefrontal cortex. And we tried to identify the molecular underpinnings of this problem. And we found specifically alterations in the function of one fundamental receptor that really is the conveyor, if you will, of multiple sensory inputs to the prefrontal cortex, and that's the glutamate receptor, NMDA. So we started looking at distribution, for example. Again, I just want to give you the high yield of results of this research, but we found discrepancies in 2 subunits. The reason why I'm bringing this up is that recently this was repeated, in fact, in humans. Again, the same group with Brunner found exactly the same changes in cells, in human cells that essentially reproduced the conditions from Brunner syndrome. So again, we see a lot of commonalities, as I mentioned. And in reality, these discrepancies lead to a reduction in function of NMDA. These are patch clamp studies. So essentially, we look at the conductance of the receptor. And we found, for example, that in MAOA knockout mice, there was a dramatic reduction of the currents, as you can see here. And generally speaking, we found that if we try to rebalance these discrepancies, these molecular discrepancies, we could reduce aggression in MAOA knockout mice. The reason why you need to remember this is simply that this NMDA will come later. And so just to keep in mind that NMDA, we found, played a role in the aggression in MAOA knockout mice. And then we'll talk a little bit more about this issue. So clearly what I told you about Brunner syndrome and MAOA knockout mice is only a tiny fraction of the problem. This is a rare syndrome, right? You don't see it typically in the population. And so ultimately it wouldn't be that relevant to the general problem. But what is relevant about MAOA is in fact that as it stands, there is at least one variant, and I should say more than one, but definitely one that is well characterized, that seems to be extremely important in the determination of aggressive behavior in the general population. This is based on this Vntr polymorphism, so variable number of tandem repeats. Essentially in the promoter of MAOA, we have this sequence that can include 2, 3, 3.5, 4, or 5 repeats. And depending on the number of repeats, this essentially dictates the activity of the enzyme. So if there are fewer repeats, we have a low activity. And if there are more repeats, we have a high activity. And as you can see, these tend to be, this low and high activity tend to be more or less equally distributed in the population, depending, of course, on the ethnicity. But we tend to see essentially 50-50, or I would say in Caucasians, it's 30-70, but generally, these are widely distributed in the population. And why is that so important? Because these variants, the variants with low activity, associated with low activity of MAOA, produce impairments in the orbitofrontal cortex. This means that, remember what I told you about hostile attribution bias? This is in fact also driven by these and the dysfunction of the orbitofrontal cortex are driven by low activity variants of MAOA. So this is quite important because you understand how this could be an issue of vulnerability. And even more importantly, when we were in Kansas, we in fact studied how this would be potentially predicting factor for violent crime. And in the correctional facility in Lansing, we found, in fact, that in the low activity forms, the low activity alleles were much more common in people convicted for violent crimes. And in fact, it was by far the best predictor of violent crimes. And this finding has been then repeated by many other groups. And it seems to this day, one of the best predictors of delinquency and violence. Why is this important, though, after all, right? What was found in 2002, and this was a seminal paper by Avshalom Caspi, he found that individuals with low MAOA, so these variants, whenever they have a history of maltreatment, remember that I told you that's a very important environmental factor, whenever these two factors synergize, so you have a low MAOA carrier that is maltreated early in life, there is a huge increase in the risk of antisocial behavior. And the reason why this is so important is that this is one of the very few gene by environment interactions that has been consistently repeated. I would say 90% of the studies and 3 separate meta-analyses have confirmed that in fact, this interaction does exist and predicts quite strongly for MAOA, for impulsive aggression and antisocial behavior. So definitely something that we wanted to study, and we wanted to develop our tools to replicate this problem in animals. So the first thing that we did was generating a novel MAOA hypomorphic line. This was 10 years ago. We did it, I would say, serendipitously, because we found that if a neomycin-resistant cassette is inserted in one of the introns of this gene, this leads to some splicing variants with a major predominance of truncated variants that get ultimately eliminated, degraded. So at the end of the day, this translates into a major reduction of MAOA, which is not zero. So this is not the MAOA knockout situation in which you have no functional MAOA, but you have approximately, we believe, about 10 to 20%. So there is a quite a drastic drop in MAOA at the level of the mRNA, protein, and so on. And these individuals, again, do not show aggression per se. So this is quite important, right? If you want to study an interaction, you need to make sure that, by itself, the genetic component is not sufficient to produce the type of phenotype. So we looked into their behavior, and we found that in terms of aggression, this resident intruder task that I mentioned here was essentially equivalent to wild type, a little bit more aggressive, but really not significantly, while the social interaction, so the social ability, was reduced. Again, remember what I told you, these variants definitely predispose to a low level of social interaction because of the orbitofrontal problems. And so this actually was pretty much in line with what we expected from these mice. But ultimately, our goal was a different one. We wanted to reproduce this gene-by-environment interaction, which took us really a long time. But finally, we developed a good translational model, and we essentially subjected these pups from the beginning of their life to a sort of child maltreatment regimen in which we alternate abuse and neglect. Abuse is saline intraperitoneal injection, which is a little painful. So again, these are not major distress factors, but they are stressful, and they are given in a pseudo-random way so that the animal can never really expect when it's coming. And of course, also daily sessions of maternal separation. When we combined these two factors, we found that our neo-mice, our hypomorphic mice, showed a major increase in aggression, starting from more or less the third, fourth week of life. So you wouldn't see major differences, at least not in aggression, in these mice, in these pups, but they would develop this high level of aggression. And this was done initially on 3 weeks of this child maltreatment schedule. And then we found, in fact, that the first week specifically was really fundamental to produce this, this outcome. And so you had to inject them during the first week of life. If you started this maltreatment from the second week of life, this wouldn't really lead to specifically aggression. It would lead to other problems, but not aggression. And the aggression that we saw, both with the 3 weeks of maltreatment, if you will, and the first week of maltreatment, were in fact appeared around postnatal day 21 to 28, I would say, so immediately after weaning, and then peaked in young adulthood. And again, this is very similar to what we see in humans, where there is, I guess, an increase of aggression throughout adolescence and then reaches the peak, if you will, in young adults. So now we had a perfect idea to study how this could go, and we had a model to study why there is this interaction. And first, we wanted to verify what could happen. And before I get into the specifics, I just want to show you a few things that really signify a few things that I mentioned earlier. And one thing, for example, that we noticed was this reactivity to a foreign animal that was in a cage. Why do we put this animal in an enclosure? Simply because we want to avoid the aggression. And so we want to really study what happens in our proband mouse without the confound of, the fight. And so we found that our animals tend to be extremely reactive against this type of cue. And in fact, there is they start doing some tail rattling, which is a major sign that it immediately precedes the aggression, and then they try to really reach through the grid of this enclosure to really grab the foreign animal. And when that happens, and I'm just mentioning this because this is now conducive to a number of new ideas that we have, this actually increases dopamine tremendously. And so I don't know how familiar you are with the dopaminergic system, but when we do this, when we expose the animal, and then at that point we let them fight, we see that already the exposure increases the levels of dopamine in the nucleus accumbens of the same amount that we see, for example, with cocaine. So just imagine how important this is to really generate habits. Dopamine is really critical for that. And in fact, now we are really studying the possibility that fights in these individuals might become addictive, and there might be a real aggression addiction that really drives the transition from what initially is just reactive aggression to a fully proactive aggression and instrumental aggression. Because of course there is an addiction component there. And this is why actually stopping pathological aggression in these individuals is extremely difficult, because they seem to really crave for aggressive cues. And so we are now using what we know about the science of addiction to really reapply this to aggression. Anyway, this was a digression, but I just wanted to give you a sense of how we are looking at this problem. When we study in general what is the mechanism behind this gene by environment interaction, we looked at many different behaviors. The first thing that we wanted to see is this specific, right? And we found that in fact, this was the only difference that we could identify specifically in these individuals, the blue column, right? The gene by environment interaction, as opposed to all their controls. There would be some differences, but they would never be specific to these individuals. Until we started looking at threat and the reaction to threat. And this was done initially with a new paradigm that we developed, in which we would put an animal, and a mouse in the start box, and there would be a little opening so that they could actually sneak out. But then there would be an anesthetized rat. The rat is a predator, in fact, in front of them. So most animals would not go out, would stay in this enclosure, in a protected environment. But what happened with our animals exposed to early stress is that not only did they get out, and quite soon, but they also spent a lot of time in the area of the rat and even climbed the rat. So just to signify how deranged their sense of threat is, which clearly points to a problem of the prefrontal cortex. And in fact, although there was no overt morphological change in the prefrontal cortex, we did identify a number of molecular alterations. And I will talk about this a little later. But one thing that I do want to tell you, again, talking about this translational idea, is that we are very interested, of course, in preventing the development of aggression. So we really looked at pups in the pups of these animals to really understand how this would work, what phenotype might give us indications about a future, I guess, a deranged trajectory towards pathological aggression. And we didn't find much, but we found one that seems to be strikingly overlapping with what we see in humans. And specifically, we found a reduction in heart rate. So almost every study, this is a huge study that was conducted in Sweden on conscript, found that bradycardia, so low resting heart rhythm, is one of the stronger predictors for future aggression, for future development of aggression. And sure enough, we found exactly the same thing. And we tested this in pups, and we found that, in fact, these individuals, these mice, have bradycardia, and this bradycardia correlates to the development of aggression. So this can give us, some sign on which we can start implementing prevention, which is a very important thing when it comes to aggression. We also looked at the molecular changes, and I told you most of the changes appeared in the PFC, but I would say the main change that we could really unravel was a selective increase of one serotonin receptor, 5-HT2A. And as you can see, this was as opposed to all other serotonin receptors, we found an increase while the levels of serotonin were extremely high, but not differently from the non-stressed counterparts. And so we had a situation in which there was a lot of serotonin, as we would expect, but also some elevation of at least one receptor, this 5-HT2A. The reason why 5-HT2A is elevated is in fact due to stress. And we found out that, in fact, it is due to one factor named CRF, corticotropin releasing factor. I apologize. I don't know why this is, sorry, somebody's, I apologize about this. Yes, as I was saying, the CRF receptor is identical, but what is elevated by stress is CRF. And we found that CRF through CRF1 essentially upregulates 5-HT2A in the prefrontal cortex. This has been documented by other groups, but it's definitely true in our case. And in fact, when we stop the CRF1 receptor, we actually rescue the aggression as well in these animals. So this is quite interesting because it might give us some insight into therapy. And then, of course, we then started looking at what would happen with 5-HT2A, and we are running interactomic studies to see whether there is a connection with the NMDA receptor, because as you remember, we really think that that's a major factor of these problems. And we found many changes that seem to be time dependent, so they follow a trajectory. But in essence, we are now studying really these problems to identify new therapeutic targets. I want to move forward to just tell you that, sure enough, NMDA receptors had quite a bit of a deficit, although it wasn't exactly the same that we found in MOA knockout mice. And we identified that there was a major reduction. As you can see, this goes from, I guess less than a tenth of aperture, so opening of function of NMDA receptors in the prefrontal cortex with a reduction of all subunits that was in fact age dependent. So it does seem, based on this data, that NMDA receptors are in fact the final effector, and they lead to a hypofunction of the prefrontal cortex. So quite interesting. And sure enough, we also identified that 5-HT2A were the mediator of this response. So we injected our animals throughout the first week as they were being maltreated with the blocker of 5-HT2A, and we found that this treatment by itself during the first week rescues the phenotype altogether. And this happens both with an injection in the intraperitoneal injection, and of course also with monoclonal antibodies for 5-HT2A on the very first day of life directly into the prefrontal cortex. Again, signifying that this elevation in 5-HT2A that we find in the prefrontal cortex of these mice is really mediating this gene by environment interaction. And sure enough, the same treatment also rescues the bradycardia in pups. So remember that I told you that the bradycardia was probably a harbinger of the development of aggression. So 5-HT2A blockade actually rescued this phenotype as well, not just the aggression, but also the harbinger of aggression itself. So this really gives us a very important tool. If we want to do prevention, we could treat kids that, for example, are at risk of developing violence and pathological aggression with 5-HT2A blockers, and we could verify whether this works primarily just looking at their heart rate, even before they develop aggression. So this is a very exciting development of our research, and we are exactly working on these, on this type of studies in kids, in orphans, for example, or kids that are in the foster system, exactly to see whether we can prevent this type of development. So far we are primarily working on biomarkers, but that's in fact the final goal. And sure enough, again, another thing that happened was that 5-HT2A was that when we blocked 5-HT2A in early life, we actually also saw a complete rescue also of the changes that we had seen in NMDA receptors. So the NMDA receptor function came back to the regular level that we would expect from these mice if they hadn't been stressed. So again, it really shows us that 5-HT2A plays a really important central role in mediating this interaction between child maltreatment and low MAOA. We are using this to develop any sort of therapy, and I want to get into the specifics because this is an additional layer, but just so you know, together, in fact, with UMKC, we started a number of studies, clinical studies, and we found one drug that is particularly promising to treat pathological aggression in adults. And that's a drug that is used for Lou Gehrig's disease, riluzole. And riluzole actually worked extremely well in our mice. And so we started testing it in aggressive patients, in fact, in Missouri. And we found a tremendous reduction with the first 4 patients, that is. After 2 weeks of treatment, we found an extreme reduction of aggression scores in these individuals that are antisocial. So we hope that this can move further. This is a very difficult study, and of course it was interrupted because of COVID, but we are planning to move forward, and hopefully this will lead us to identify new therapies for pathological aggression. So just to give you a general summary of this part, and then we'll talk a little bit about sex differences before I close. I just want to recap what we found so far. We identified that low levels of MAOA lead to high levels of serotonin, right? Remember, particularly early in childhood. But when the individual is subject to child abuse or neglect, there is activation of the CRF system. And ultimately, this leads to an activation. So on one hand, you have a lot of serotonin. On the other hand, you have more, I should say, of 5-HT2A in the prefrontal cortex. When these two mechanisms synergize, you have activation of these receptors, and this ultimately leads to changes in NMDA signaling, and so a progressive reduction of the function in the prefrontal cortex, and therefore impulsive aggression and pathological aggression at large. Then, depending on the experience, this might move even into proactive aggression. In a few minutes, and I really don't want to take more than 5, I just want to tell you a little bit about our studies in terms of sex differences, because this problem seems to be more predominant in males. If you just look at the statistics, it's very common that men rather than women are involved. And to some extent, I also told you MAOA is X-linked. So that should be a factor. But in reality, even when we factor for that, we still see that there is a predominance of involvement of males in this delinquent or violent acts. And in fact, what we found, what we have in the literature, in the clinical literature, is very confusing on females. You have some studies that show that, yeah, there is a similar gene-environment interaction, some that really show quite the opposite. And what we found was a very interesting picture, because in reality, we found that in diestrus, there was in fact a similar, albeit as lower, increase in aggression just in a gene by environment, but not in estrus. So it does seem that hormonal factors really drive the score here. Depending on the cycle of the female, we have a higher or equal proclivity to engage in aggression. And of course, this we are very interested in this in neurosteroids, and we started looking into these problems. And we looked at a number of enzymes, but one that is of particular interest to us is this enzyme here, 5-alpha reductase, which is also, important in processing progesterone, which is very high in diestrus, and its metabolism into this this factor here named allopregnanolone, which is a very important stress factor. And so without getting into too many specifics, and we can talk about it later if you will, essentially we looked at 5-alpha reductase in these animals, and essentially we found a major increase in one of these 5-alpha reductases, the second form, 5-alpha reductase 2, in the nucleus accumbens. And we are now running a number of studies that seem to really show that the involvement of this enzyme might be particularly important in the acquisition of aggression, particularly of proactive aggression and of dominance. We've already published a paper in which we showed that this enzyme in particular seems to be extremely critical to really for the acquisition of dominance and for the acquisition of proactive aggression. And so we are now applying these ideas to our model that I showed you. And of course, we are running a number of studies on the profile. And yeah, we keep seeing a number of alterations that occur in our mice of gene by environment interaction. There is a clear increase in some forms, and particularly in the products of 5-alpha reductase. Yeah, sure enough, I can probably skip this point. We looked at one of these factors, allopregnanolone, so remember the metabolite of progesterone, and we found that essentially when the individual is stressed, so remember the social stress that I showed you before, right, with the foreign animal in the cage, and there was this huge increase in dopamine, and we want to understand what is causing that, right? What is the stress factor that leads to that? And it appears that in fact allopregnanolone might be the factor, because most of the environmental changes that tend to increase aggression in these individuals tend to also support an increase in allopregnanolone. So for example, in this case, right, whenever there is this social stress or sleep deprivation, which is another major factor, they all increase allopregnanolone. And this increase in allopregnanolone is particularly interesting because we think it is mechanistically a fundamental driver, because when we actually interrupt the signaling or the synthesis of allopregnanolone, so whenever we give a blocker of either 5-alpha reductase or the receptor of allopregnanolone, we actually see that this aggression in response to stress tends to go immediately down. So this tells us that perhaps there is another alternative way to tackle the problem, and it's really to intervene on the biological factors that drive to aggression in the presence of a social stress or in the presence of another stress that tends to increase aggression. And allopregnanolone might be really an important factor there. So in conclusion, yeah, MAOA deficiency, as you saw, and I hope I convinced you, increases the vulnerability for pathological aggression through alterations in environmental adaptability, particularly due to the prefrontal cortex and the orbitofrontal cortex, I should say. And then, of course, this mechanism, mediation, is based on the activation of 5-HT2A receptor in early life. So that's a very important target that leads to hypofrontality. And there are stress factors that might really be critical in promoting the immediate spurt of aggression that these individuals experience whenever they're under stress and leads them to violent actions. And so we could actually use this target target to intervene significantly in reducing aggression in individuals that are already aggressive. And so whenever we cannot prevent, we can probably have some new drugs to treat this, this problem. In closure, I just need to acknowledge, well, this is in fact our building here. It's in the midst of a park. So whenever you guys want to come to Utah, please come see us. This is a very nice area. But so I need to, of course, acknowledge a very large number of people, collaborators all across the world, really, that have helped me quite a bit with achieving some of these results. And of course, my lab members that are still active on this project, and the generous funding that we received and hopefully will keep receiving, knocking on wood, from NIH and other foundations. And I really want to thank you for inviting me to talk about this topic. Thank you very much. And I'm happy to take questions.
Thank you, Dr. Bortolato, for such an amazing and stimulating seminar. And again, the best research raises the most questions. And I have several.
So let's see if I get any.
I have several. I'll just ask one.
Go ahead.
So I kept thinking about the use of MDMA, ecstasy, as a treatment for PTSD and perhaps aggression. But it seems counterintuitive. Your research seems to go against, because in my understanding, ecstasy increases serotonin. You might expect more aggression.
It is true.
So can you reconcile those things?
Oh yeah, without a doubt. The issue here is not so much as serotonin, yes or no, but it's serotonin when. The development, so as I told you, having these very high amounts, high levels of serotonin early in life is also what we think is one of the factors that predispose us to autism. There is a reason why these animals are autistic-like, because it essentially, it generates such a florid arborization in the prefrontal cortex that the prefrontal cortex becomes unable to process social stimuli. So too little or too much, in fact, particularly in early life, can lead to very similar final outcomes, because ultimately it's the cortex that is unable to process stimuli. So if you try, for example, to block MAOA in late life, right, you give an antidepressant, yeah, you don't get aggression. Of course you don't, because that's past the phase of morphogenesis in the brain. So it's really important that this is genetically driven, or at the very least, I would say, there are some experiments, in fact, that replicated this with blockers of MAOA early in life. And so if you do that early in life, yeah, you get aggressive outcomes. But it needs to be specific to the morphogenic properties of serotonin, and of course the fact that particularly in early life the levels of serotonin are very high no matter what in the brain, because they are so important in shaping circuitry. So at that stage, if you have a blocker of the enzymatic degradation, of course this makes a huge difference. So to just give you a more direct answer, if we had, for example, hallucinogens in early life, I don't know. I think there would be a very good possibility. I mean, MDMA is slightly different, but say, for example, psilocybin or any LSD in early life, yeah, it's possible that these individuals might become very aggressive for the same token. And maybe also a promoter of serotonin levels, even though that is slightly different than blocking the metabolism. But yeah, by all means. But I think really what is critical here is the timing.
It seems like you've got that question before. So I'm going to put on my...
no, it's a question that I posed to myself a lot, right? Of course.
I'm going to put on my NIH grant reviewer hat now and say your MAOA knockout is pretty conclusive, or at least it's correlative for sure with increased serotonin early in life. So then the question becomes, if you infuse it early in life, infuse serotonin, have those experiments been done?
So as some of that, right? But the problem is that MAOA is very effective. So yeah, to some extent you can, but that will still be degraded. So the issue here is to get accumulation of those levels. And that level of accumulation you really get only if you stop the metabolism altogether. You can certainly increase serotonin, but as long as MAOA is able to degrade it, you still won't have that type of outcome.
Awesome.
If it makes sense.
Dr. Hirata, go ahead.
So first off, amazing work. Like, I mean,
Thank you.
The translational aspect of this absolutely blew me away. And I've got some of my students in the audience, and we've been texting back and forth. So amazing.
It's very kind of you to say.
So we work on the gut and, host microbe interactions. There's a lot of, serotonin, obviously 5-HT generated in the gut. Do you have any evidence of the microbiota as an environmental factor playing this? Because I mean, I'd be surprised if there wasn't some component involved.
It's so funny that you ask, because one of the 3 grants that we just applied to is exactly that.
Awesome.
We are collaborating with Toronto to get feces, and we will transplant our mice with feces from violent individuals if they give us the funding, which is that's a gift. But so I will say this. If you want my opinion, I absolutely believe that's a major factor, because one side of the story that I didn't present here is that, there is quite a bit of involvement of cytokines. And if you just want an interesting clue, it does look like propionic acid compounds the NMDA deficits that I told you about. So my take is that by changing the environment of serotonin in the gut, low MAOA can also promote the formation of these short chain fatty acid that then reach the brain and compound NMDA problems. So no, I don't think that that's a causal factor. I don't have any reason to believe that that's the case, but I definitely think it's a major compounding factor. And that's what we want to study. And I think it's driven partially by cytokines, particularly IL-1 beta, because it does have a very important role in the hypothalamus. And the other is, in fact, propionic acid, which is mind-blowing and so interesting to me. But so we hope we'll get some money. I guess you'll have to ask me in a few years.
Okay, awesome. I look forward to hearing those studies. I think that's going to be really, really cool.
Thank you. I'm happy to take any other questions if you guys have them. I don't know. I'm really terrible at this. Dennis, I think, right?
Dr. Wolf, go ahead.
I don't know that I can't hear you. I don't know why that is.
Oh, I'm sorry.
Oh, yeah, now I can.
Had myself muted at a second place. So I found what you said here fascinating. And so the things that are curious to me was to what extent things like this might be plastic. I mean, it seems to me that in reality, we're not going to be able to lock up young kids that were mistreated at youth and force them to be treated. I mean, we're having trouble treating adults right now. And so I'm curious how you see this coming out in the future, and to what extent can we actually retrain? I mean, to what extent can intelligence overwhelm instinct?
You nailed it, right? So the idea here is, of course, I mean, before we get into the point of preventative therapy, there needs to be a lot of research on this. And so our first question is whether we can retrace the same type of development in kids that have the same type of origin. And so that's what we're proposing to do. To what extent can you intervene? I think the earlier you intervene, the better. Now, the question with this is whether there might be some forms of microaggression that are reported in kids, and some of them actually do engage in this. And so that becomes a completely different issue. If the behavior is the harbinger is already partially pathological, so for example, some sort of an antisocial behavior, then the question is, can we intervene there? Is it a good option to use, for example, 5-HT2A blockers? And here I'm talking about olanzapine, to give you that's quite a potent, not a very clean one, but still. Those are used in aggressive children. Now, the question, of course, becomes whether it is an issue of therapy or an issue of prevention. And I think you're absolutely right. The issue of prevention is much more complicated. But that said, if we identified there are biomarkers that overlap between our mice and patients, and we identify that, in fact, this gives us a sort of ID card of risk, then the question needs to be, can we start at the very least behavioral treatment? And the literature suggests that if you start early, there is a very good chance that you could counter at least some of those developments in the prefrontal cortex, because they are fueled by the consistent loop of you having this social interpretation bias, and at the same time, so engaging in aggressive behavior that might really build up. So clearly some behavioral intervention, clearly some psychosocial intervention is likely to improve this. Whether we can test it in mice, that's a different story, because you're really scratching the edge of what can be feasible in animal models. But the idea here is, so I want to, I guess, rephrase what I said about long-term. We definitely hope that at some point, identifying elements of risk for a trajectory towards violence and antisocial behavior, which ultimately leads oftentimes to conviction in prison. So it's quite an important issue, might at least inform behavioral treatment. And so what we want to really get is a sort of list of factors that might help us quantify the degree of risk that this individual has to become a potential violent individual or an antisocial individual. And so whether then we can really discuss treatment as a prevention, yeah, I agree with you, particularly when it comes to pharmacology. It would have to be a drug that really has next to no side effects, and as we know, that's very hard to find. So that is a complicated ethical question, and I don't know that I would have the answer right now. I agree. I'm not sure that that's something that we can really ask. But what we want to have is really identifying, okay, what are the factors that really allow us to quantify and measure the level of risk that this kid that has been maltreated might really develop aggression, because as you know, many of those don't, and just go into depression or other types of outcomes. So that's what we really want to distill from this type of research, at least in its translational application. Did I answer your question?
No, thank you. I mean, I think it's fascinating, but it always leads to these complex questions of what do we do with what we know?
Really, and I'm sure that you caught that there are many ethically interesting repercussions of something that I said. For example, that genes drive so much that are such an important factor for violent crime. That is already mind-blowing if you think about it. These are really complex questions, and I don't know, that goes beyond the scope of what I do, I guess.
I can hear my father now. Jeff, look at you. Such gene-driven behavior. It's amazing.
Well, you can always blame him.
Yeah, it's all his fault, no question. All right. Any students left in the room? Here's your opportunity to ask questions for Dr. Bortolato. I see a hand. Dr. Agbas, go ahead.
Hi, good afternoon, Dr. Bortolato. Thank you for sharing your science with us. It's very interesting. Some of the prison systems in some countries, operating the sex offenders by castration, they remove the testicles or doing the something. Does it affect on the MAOA levels, or further increase or decrease the aggression?
So Dr.
The guy lost something, so I think it just induces the more aggression.
Did you catch the last part of the talk when I was talking about finasteride?
Probably I missed that part.
So anyway, yeah, we're looking into that, but not necessarily because it acts on MAOA, but it acts on the hormonal system that also interact on this. So finasteride does reduce aggression, which is quite mind-blowing because, and I didn't discuss any of it, but this implicates that it's not so much testosterone that drives aggression, but it's derivatives, and so DHT. And that's, in fact, another chapter that I didn't really talk about, but we are exploring. So it does reduce aggression, and we do have the proof that when we, for example, when we castrate our animals, we have a tremendous reduction in aggression. Not a complete ablation, but we do have a reduction. But what seems to be the cause, based on our studies, is not testosterone, as pretty much everybody is assuming, but it's derivative. So DHT, which is why we're targeting the metabolism of testosterone rather than testosterone itself. And we are now performing studies to see whether we can rescue the aggression, restore the aggression just by implementing DHT as opposed to testosterone. And so I think that the data that we have strongly support that possibility, but we're still operating on that.
Which means that they can get the DHTA treatment not to lose.
Well, I don't know that I would give it to criminals, right? Because that would increase aggression, but conceptually, that's what we want to explore.
Okay, thank you.
And there's direct evidence from the farm. I used to raise cattle, and if there was a bull that was uncastrated on my farm, my son would go nowhere near it because it was just so aggressive.
Yeah, I mean, I will tell you this. Aggression is also present in females. So it's not absolutely just that. What I described about the cortex stays, but it is clearly a determining factor. So it is one of the many factors that interacts in the big picture.
And when we castrated those bulls, they became way more docile.
Yeah, and that's exactly how it goes.
No question. Okay, any more questions? Okay, if not, this is the time I say thank you to Dr. Marco Bortolato.
Thank you for having me. It was a real pleasure. Perhaps in the future we'll do it in person, hopefully.
Yeah,
right?
that'd be great.
Thank you so much.
I'll send you the links to your talk and you can listen to yourself.
Thank you. That's very nice.
Okay.
So it was a pleasure to meet you all, and I wish you the best, and I'm sure we'll see each other again. Have a good one. Bye-bye.